Just wanted to welcome everyone back if you've been here before, and welcome to the community if it's your first time here. I think it's a good resource for people who are trying to work on OGM or solve problems or are just interested. Usually we try and have recurring meetings every quarter, but depending on how busy everybody is, that can occasionally be a little longer. Usually we'll have a call for cases, so if you have cases that are interesting coming across your desk or problematic or educational in some way, save them. It's a good forum to share your cases and get everyone's thoughts or just show people how useful OGM can be. I see we have a lot of representation from across the country and internationally, including some folks from Nano. So again, welcome everyone. I wanted to send a quick reminder of a listserv we have set up for over a year now. It's been really quiet. So this is the address, and I can send it out to everyone in attendance after the meeting if you want to sign up onto this listserv. If you have questions on OGM or things you want to chat about with people who are using it in this community, you can just send an email to this address and it'll push to everyone on that listserv. And that may be a good forum to asynchronously discuss cases with a bunch of people and see what people's thoughts are, or even just chat. So the agenda for the rest of the day is lightish. We have first up a discussion of VIA and its applications with Dr. Susan Crocker. We have a couple of people who are interested in presenting some interesting cases, so I wanted to leave a fair bit of time for that. I know people have worked hard on those. Those are always fun to walk through and discuss. And then if we have time, we have some data from our around 1 year of experience using OGM for acute leukemias, which we presented at ASH recently, sitting around. So we're happy to share that, show the added yield of OGM and acute leukemia specifically, again, if we have time. But I also want to make sure we leave time for open discussion of problems people are having or things people want to focus on. So I know there's been a lot of interest in external quality assurance, which is obviously a problem for everyone. So if we have time, we can definitely talk about the situation there. Right now, from our perspective, we're still working on that with our privacy office, but we can chat more about that when we get there. So with that, I'll pass this out to Susan Crocker. One thing I wanted to announce at this meeting is that Dr. Crocker will be taking on one of the co-chair positions of this community of practice beginning in January 2025. So we're very happy to have her with her wealth of experience in OGM. So I think you should be able to share your screen, Susan, but if you have problems, please let me know.
Thanks very much, Eric. Thanks, Adam, too, for helping organize all this for this session, too. Okay, thank you very much for the invitation to also speak today. I'm grateful for an opportunity to just lead in the session a little bit. I've inserted a couple of poll slides in here too for our benefit, and please have some patience with me as I'm trying to see if I can get this to work in real time and live. So hopefully that works, that I can see your data as we go as well. Tried 2 different approaches for embedding, so hopefully it works. What my hope is today is to, everybody in this call is familiar with the advantages of this new technology and some of the background and how it works. So I won't get into the weeds of that at all. I'm just strictly going to focus on a little bit of a quick overview of what it is that we've done at our center, how we've incorporated it for the people who are on the call who may or may not be aware of it, and how we've realized some efficiencies by incorporating it into the clinical service. I'm going to illustrate not only the improvements in care using optical genome mapping very briefly. This is just a quick 20-minute talk. It's going to be mostly moving along to the cases and then our group discussion, and also review some of the interpretive improvements that we realized by integrating VIA into the clinical service. So as I said, I'm not going to get into the weeds of using of the actual overview and the process for processing optical genome mapping. I'm going to make an assumption that everybody is very well versed in the 1.5 million cells or low input that some of the newer workflows are trying to push the limits of what can be used. Are all the considerations around ultra-high molecular weight DNA extraction, the labeling, chemistry, and running it on your different instruments, be that Stratus or Sapphire, the various bits and bobs of software using Access and VIA, and the advantages of how you use those for reviewing cases. Though we're going to talk a little bit about specific to some of the things around VIA, and then the ability to align and visualize and analyze that data using those two softwares. We certainly have been using and doing optical genome mapping for a bit of time, since 2021. So we've gone through many different iterations of different software versions, updates, different chemistries as well, different chips as well. We have at our center 4 servers. We have one Sapphire server, and we have attached to one instrument, and then we have one Sapphire and 2 Bionano compute servers attached to the other instruments. So it allows us a little bit of flexibility to be able to sandbox one of our instruments, so we can trial things before we bring it live onto our instrument that we are running the clinical cases on. And we've been using optical genome mapping to study it in a variety of different ways for different uses. We have different approaches working with different stakeholders for the way that we look at the data, the reporting templates that we use on it, the analysis templates and decision trees that we use in VIA. So, and they differ slightly depending on what it is we're doing. So some of the approaches I'm going to go through in terms of focusing on a little bit about the advantages that we saw with failed bone marrow cultures, and which we still use optical genome mapping in that instance. How we initially were able to get one of our 2 Sapphire instruments on site using some of the funds associated with a Pfizer grant for targeted for acute myeloid leukemia. And how we're using it for specifically CLL. I'll talk a little bit about that quickly in the presentation as well. And then I'm not going to get into the weeds too much about the infertility, recurrent pregnancy loss, premature ovarian failure work that we're doing, but I can certainly talk about that at a future community of practice talk in the new year. So this is just a quick snapshot that I've presented before at a series of meetings, just to show the advantage of using this technology. This was a quick overview of some cases from 14 cases in a 12-month period that had low mitotic index and no results that we'd used optical genome mapping for. In bold were the ones that we had additional abnormalities that were described by optical genome mapping, and certainly in those ones that we had absolutely no information on, it certainly has proved its utility over time. And as soon as we actually were able to get the test up and going, I know Adam has shown and shared several times about the shocking improvement as a cytogeneticist in terms of the level of resolution that we achieved once we started integrating this technology into our practice. And certainly for us as a center that had reoccurrent bottlenecks and issues and problems with HHR issues over the years, and we were in a state for some of our routine bone marrows that were non-acute of being in a fairly embarrassing, I'll say that, and right, full disclosure, and frustrating situation with bone marrow analyses being well outside of what would be acceptable, or even assist the clinic in any sort of way for management of those patients. The ability to incorporate optical genome mapping into the lab allowed us to pivot several resources so that we could streamline some of the way we targeted the FISH testing for these patients, as well as incorporating it in with a variety of other different steps that we have done to lean the service a little bit. So certainly it has proven its utility that way as well. Now, the way that the data interpretation is done, certainly most of us are aware of the fact that we've set several of the metrics for both the pre-filtration according to the recommended Bionano specifications. We've also, the VIA has allowed us to improve on the artifactual and polymorphic variants as well. We'll touch lightly on that as well. And we've also had various criteria depending on the referring diagnosis. for various CNVs. This is SV, so this is an older, derived from an older paper, which the reference is included here, but certainly the workflow holds true for what we're talking about today. The advantages that we certainly were waiting on before we started moving this into clinical practice was the nomenclature, which thank you, Adam, for being on the call. Thank you, Jean, for being on the call today. Certainly their hard work with the optical genome mapping community made it possible that we had the language to be able to include in our reports and to share with each other across the country. It's wonderful to see the incorporation of ISC, the OGM nomenclature in the newest version of ISCN. So that was something that we were hoping to have, as well as the framework for the validation studies that also were out and published recently as well in the past couple of years. But the VIA software really makes the data a lot more accessible to the clinical lab, not even from just the perspective of the clinical laboratory scientists, but also from the level of the MLTs and our variant interpretation technologists as well, to make the volume of the number of calls a size that's manageable within the workflow that we have with the volume of samples that we have. And certainly the various criteria that we'd use from ClinVar and ACMG for the various tierings or levels of pathogenicity. Certainly the incorporation of that would follow suit with any sort of other variant interpretation you'd be following for sequencing results, except for in those cases where we use a pared down fish-like panel approach for some of the results that come out for optical genome mapping. So this is the first poll, and I'm going to see if people can use this to log in and see it. And I'm just going to see if I can use this to log in here. People can log in. I'm going to give you guys just a couple of minutes here. See this just a second. Just to log in.
Sorry, while everyone's doing this, I thought I should mention, if you guys have questions that come up in the meantime, you can always hold them for later, but if you want to enter them in the chat, I'll keep an eye on that as well. We can discuss whatever lands in there.
Okay, so thank you, everybody can visualize it now. So it looks like a third of people are using VIA, and so 36%, 38%. And then 62%, 63% are not. So the next few slides will be helpful, I imagine. So we'll talk a little bit about that. And certainly we can incorporate in the community of practice, community of interest, further discussions around it and review and some of the different approaches to have a more fulsome discussion about things as well. Okay, so here's the second poll. See how people answer this one. So about building your own decision trees. For those of you that third of people that are actually using VIA, 50%. So about 44% of people are using their own decision trees. I guess the other, so I'm presuming those of you that are using VIA right now are using probably the ones that are already pre-built within VIA, which should have been another poll question. But you can please feel free to use the chat to enter in those answers to that particular question. Okay, so this is good that I know that I'm talking to about 60% of the people on the the call to just try and give a quick overview about what are the advantages of VIA, and give you a couple case examples around that within the remaining time. I'm also going to introduce Henry Wong as well, who's going to explain a little bit about what the decision tree is and what the advantage about that is as well. So I'll be introducing him. Hi, Henry. Put your camera on. That's good. As well, to explain who the heck Henry is for those of you who aren't aware who he is. Just a second. So, okay. So simplifying and customizing clinical interpretation. That's the crux of what VIA does. VIA allows us to build a knowledge base internally. We're also in discussions about the potential for sharing between centers the knowledge base, and possibly we could build off that as a community of practice, as well as an item for further future discussion. It allows us to build these things, as I mentioned, called decision trees that allow us to customize our approaches in terms of making the data manageable that comes out for review. And also, I mean, certainly VIA also has the ability to, I mean, it was built off of biodiscovery, so it's very good at copy number calling, as most of you are probably familiar with using it for next generation sequencing, and you can certainly input NGS data to layer in some of the different types of technologies all together within that software as well. You can also do automated classification as well. Allows you to go and put the various categories for the various variant interpretation calls that you would like to make, and it layers in all the databases to aid curation. You're able to see, visualize the data and some of the tracks all within the same field of view, which is extremely helpful when you're reviewing the data. So when I say simplifying variant interpretation, I'm going to show you, for those of you who are well versed in reviewing optical genome mapping data through Access and have spent time, and I know Adam and Eric can both sympathize and empathize with me when I say that those of us who were there in the very beginning, patiently going through and manually curating each of the calls and going back to look at the molecules through using Access only, and then not having a knowledge base built. Certainly, it's a time investment per case, I will say. So perhaps you can appreciate that there are multiple pages of variants here. Each of these were required a review of, and then a character classification to be assigned on each of these calls. When we do VIA, and Henry's going to walk through a little bit about what each of these fields are and how you populate these with the decision trees as well. You can get metrics at a glance using VIA. So it allows us to get some quality scores, right, as well as we can see which sort of we can see which analysis pipeline we've used on it for a particular sample. You can see the sample type, so a little bit of other metrics around which of the various of the decision trees you've applied within your analysis as well. The status and where it is in VIA, if it's being processed or not, is also would be found here as well. And then you can see the various classifications, which can be some of these are already custom built within VIA from the BioNano team, and then you can see things, I guess, at a glance, like from before you actually even open up and go into things. And when you go into the actual view within VIA, you can actually see, you can code each of the classifications to have it color coded if you'd like to cut and see the calls associated with your list here, and your decision trees can clean up how much or how little you want to see of the data, because our stakeholders each have different levels of tolerance of how much information they actually want coming out on their cytogenetics reports at this particular time. So we've worked with them fairly closely to try and map out some of those. And certainly you can see here that the artifacts are fairly well labeled once you've made your classifications for that at your center. So things like, and also show you another important thing that is improved with VIA as well, This is just one particular case that had two predominant clones. One of them has two clones, where the predominant clone has a loss of the long arm of chromosome 13. And which you can see here, this is the smaller clone, less predominant clone, which had an isodicentric 17, chromosome 17 right here. And it also had a gain of a chromosome 11. When we first did optical genome mapping to take a look at this case in Access, and this is tiny on my screen, hopefully it's a little bit larger on yours and you can visualize it. We could certainly visualize the del13, and it was supported by both a copy number call, and you actually had fusion maps that go across where the SV call was actually supportive of this loss of chromosome 13, but we couldn't actually see the loss of chromosome, we couldn't see the isodicentric chromosome 17, so the structural rearrangement and the loss of material from chromosome 17, and we couldn't see the gain of chromosome 11. So when we load the data into VIA, the algorithms are slightly different, especially for the copy number calling within VIA, as I understand. And I know Alex Hasty is on the call, so in the discussion, he may be able to fill in a bit more about that as well. But you can beautifully see the isodicentric 17. So you can see not only the gain of what material is incorporated in that, you can actually see the breakpoints in the reunion within the abnormal chromosome 17. So this shows the breaking and the refusion there. And we can see the gain of chromosome 11 as well for the lesser clone. The other thing is the improved curation, as I said. So we were able to go and build and improve our internal database to customize it a little bit for curation for different disease types, so we can trim down things we have for the referring diagnosis of chronic lymphocytic leukemia, CLL, to a specific calling algorithm that our stakeholders wanted to see. And so, which vastly improved our ability to go and quickly review the data. But obviously, this is not a solo adventure. There have been lots and lots of wonderful people that have helped to support us moving forward and advancing so quickly at our center. And one of the key pivotal people associated with that is Dr. Wong, who's our assay development scientist, who has helped us build and customize our decision trees. And so I'm going to push it over to him to walk through some of the slides and discuss it.
Thank you, Susan, and thank you for the organizers for the opportunity to speak today. You guys can hear me okay? Okay, great, thanks. So some of you who are familiar with VIA, and for those who aren't using VIA, you would have seen in previous slides from Susan's that at the bottom, especially where there are different categories of SVs being labeled or classified in VIA, they all kind of look different because is a very customizable feature of the software. So today I'm not going to focus on all the different features of VIA, but instead I'm going to talk to you about the pre-classification features of VIA, how we utilize it, and how we customize the decision trees architecture that Bionano has provided as a base, and we kind of leverage that to build and customize some of our own wishes into the tree. So what Bionano has done with the decision tree is a very hierarchical logic steps that it goes through. So if you would advance to the next slide for me, please. In the top here, I'm just showing you a very small glimpse of what the code or logic looks like. You would leverage the there are two different things that Bionano uses. They're called regions files, and these regions files are essentially bed files of your regions of interest. And so by providing regions that you either customize your own or from Bionano, you can set up these arguments and string them together using Boolean terms such as and, or, and if, and provide some quality metrics if you're familiar with them, and have the software review the SVs that's detected and then go through the decision tree to kind of bin them as artifactual calls, real calls, tier 1 calls, etc. So the basic architecture of the binano decision tree that's provided, such as the AML pre-classification decision tree, is that it first goes through these disease-specific SV calls. So for example, it will go through the gene fusions according to disease specific, such as AML regions, and then it will go through and review the deletion calls with and without regions files, et cetera, and then it will do the same group of calls through the pan-heme region files, and then it will proceed to the next step where it will compare the SVs and the CNVs to the civic genes. And then the next step it does is that it looks at the CNV detections from your case history. So this would be through the knowledge base. And then finally, it will compare against the DGV entries to decide whether the call is valid or not. So from going through all these logics and call and binning, we were able to really trim down from dozens and sometimes even 100 calls per sample if it's a really, really interesting and complex case to much more manageable handful of calls, maybe in the 10 to 20 lines. And so it really cleans up the data and groups them accordingly for you. It's important to also stress that the classification based on this decision tree can be adjusted as well. So an analyst would go through these and they can change a classification call, and all the changes are logged so that the next analyst or director can kind of follow through the work and decide whether the change is valid or not. And as well, in the first presentation of this data, all the pre-classification comments is provided for you, so you can go through the line and say, oh, this variant is classified as this based on this line in the decision tree. And so we've been finding this helpful in helping our analysts to go through the cases and give them a really logical way to systematically analyze the data for us. So I'll stop from there. If you guys have any questions, feel free to reach out, or we can meet at the next user group.
Thanks very much, Henry. So I'm going to wrap up here just to say that I think we can all appreciate that optical genome mapping has allowed us to see the genome in a whole new way. Excited to see how everything advances, especially given that the United States seems to be ramping up to catch up with Canada, which is a rare thing, and very excited about as well in terms of our successes up north. And want to give thanks to everybody who's actually helped to contribute to us getting to this point. And that's a long and lengthy list for sure. So we certainly have many more exciting things to come. I see that Melanie has her hand up as well. So I'm going to stop my presentation here.
Yes, so I have my hands. It's not a question, it's more of a comment. So I'm happy to let people who have questions about your presentation, Susan, go ahead first, and then when all is said and done, I can comment.
Yeah, for sure. Thanks, Melanie. Eric, I'm going to hand it back to you. So.
Hey, Susan and Henry, that was a very helpful presentation. We are still in the dark pre-VA ages at BGH, so I can attest to the fact that it is, I like the word you used, an investment time per case. Are there any questions from the broader group?
And certainly if everybody wants to work on decision trees or have specific discussions around decision trees, I would go and ask if people wouldn't mind just putting that in the chat for us to have be able to incorporate in for our next agenda for everybody for next time too.
And I think we've talked in the past about common gene lists, but I think now that VIA is here and useful, common decision trees and ways to tweak that to individual sites makes a lot of sense, where rather than rebuild from the ground, you can tweak to what works for you.
Yeah.
I see Ken Craddock has mentioned in the chat, can you comment on the current availability of tech support from BioNano for this?
That's a
Right.
really, yeah. Yeah, no, I don't mind answering that quickly. Just to say, a really great question. We of course had the advantage that a lot of what we needed to learn and build took place before the last restructuring and changes within some of the organization for BioNano. Certainly, I know there's been a few more bits and bobs that I'm sure is concerning for the community as well. So we were already built for a few things before requiring additional supports from BioNano, but even with some of the questions that we've had and problems that we've encountered, they've been very responsive from our perspective. I know that may not be shared across the country. We've had a few blips here and there where things were slowed down a little bit, but so far, despite the restructuring, we've been okay. And I don't know if that's also shared across or not, but yeah.
I can say from our perspective, we haven't gone through the VIA process yet, but we are about to, so we can let you know how that support is. I know the VIA has always kind of been a separate entity, at least from personnel, at least from our understanding, but our non-VIA needs have been still met. So I can say that, at least again, in Vancouver's experience, it's been bringing the support we need. One other question I had was, how long did it take you guys to build your decision trees and get comfortable with how that works? Was it iterative? Was it long?
I can take that. Yeah, so we started off with the CLO indication, and we modified the BioNano decision trees with our own regions files, etc. So absolutely, the process is iterative, and it seems to be that the more we learn about the capability of the decision tree, the more we kept going back to ask how we integrate this and that into the decision tree. And Lily at BioNano was super helpful and patient with us, and eternally grateful for her expertise in getting us set up.
If I may chime in, sorry. We have started using VIA. We are on, I think, on version 6 of the decision tree for myeloid. I will be honest, we started off using the template that was provided by BioNano for myeloid, but found that at least in our cohort, which is includes a lot of pediatric cases, VIA was not calling what we needed, because I think the information that is in there, like what has been preset by BioNano, I think is mostly geared towards adult, and certainly not all gene fusions partners are in there. And so that's one of the, I would give the advice to anyone who's looking into creating their decision tree. I would say you need a very strong, very complete list of genes with actual mechanism. If you just have a list of gene in a certain disease, the problem is that you'll be calling any sort of alteration in that gene, but the mechanism may not correspond to the disease mechanism, and you end up lots of false positive calls. And
Agreed.
so I think you need to have a gene list. You also have to have a list of mechanisms, knowing that some genes have different mechanism. And for genes that have multiple partners, including KMT2A, you have to go through the painful process of entering each and every possible combination of fusion partners if you really want this to get called and pre-classified as a tier 1 abnormality. And so as I said, so we've gone through a painful process. I think we're now in a version where we're comfortable that things are being pre-classified as tier 1, and there's very little manually reclassification that has to occur. The other thing I would say is that you can include quality filters based on the quality score in VIA. that is recommended. I think the threshold by BioNano is a score of 20. I will say in our experience, this is too high because we are actually missing some well-known fusions like PML-RARA. It's calling one of the fusion, but not the other one, not the reciprocal fusion. So if you include an SV quality score, I would caution people either to revise the threshold or put it in your decision tree, so that in a logical place where you're not filtering out calls that are a bit lower quality for whatever reason, but are actually the driver of your. So that's why we are on iterate. It took us that many versions that we've been working on this for months. And I will say for AML, there's a great start. You can you need to tweak it in our personal experience. We've been working with BALL, so we had to start from scratch. And
Yes.
had to enter every single possible partner for KMT2A based on that publication from 2020. So it's hours and hours of labor. It is an investment upfront, but once you've done it, you can tweak your tree to really pre-classify things in a very efficient way without too many false positives. Being mindful of the fact that sometimes when you have 3 ways, you have to dig out one of the partners out of the low quality SV bin, right? So it's not, there will always be some manual review required. That's at least our experience.
Yeah, so which is why I think it's very important that we probably have some disease-specific discussions around decision trees for the community of practice. So certainly in our experience as well, we don't use the BioNano provided decision trees. We adapt them and build off of them and modify them. So that holds true. That holds true for our center as well. And I do think it would be useful to have a discussion about the various approaches. We've used a slightly modified approach for trying to tackle it as well, for making sure that we don't miss any calls. We used a slightly modified version from that. But so in the interest of time, because I don't want to occupy all the time, I'll hand it back to Eric. Thanks for that, Melanie. That was very useful feedback to the group. I appreciate it.
Yeah, if there's a meeting organized to discuss decision trees, I'm happy to contribute.
I think with the user group meeting that we're hoping to also host in the spring sometime, we'll also try to make sure we have a workshop for that too. So thanks.
Great, thank you for that excellent discussion, and definitely things to move forward with. So I think we'll be reaching out in the new year to chat about these problems and others. That was our experience as well. We haven't used VIA, but seeing the gene lists, they didn't work. So in the interest of time, want to make sure that people who prepared cases have a chance to present them. I'll pass this along to Dr. Fatma Al-Balushi. She's a clinical fellow working with us at Vancouver General and has been getting a bit of a trial by fire in OGM. So Fatma, if you're able to share your screen, please try to do so.
Okay, before I start, I would like to thank the organizing committee and Eric for this opportunity. I will be sharing a couple of cases that I have seen through my training in VGH. The cases are around acute myeloid leukemia. I will start with the first case, 32 years old, presented with pancytopenia and circulating blast. The bone marrow examination showed more than 20% blast, and immunophenotyping confirmed these blasts to be myeloid in origin with monocytic differentiation. Karyotyping for this case showed an abnormal male karyotype with the translocation 19. You can see that there is reciprocal translocation between the long arm of chromosome 1 and chromosome 9, this resulting in a partial trisomy of 1q and loss of the terminal region of 9q. This was seen in 3 metaphases, and the other 7 metaphases had a normal male karyotype. Optical genome mapping for this patient showed translocation 1011, resulting in KMT2A and MLL-T10 fusion. This was seen in 86% of the cells. Having a closer look at this 1011 translocation, you can see here the molecule showing a translocation involving MLL-T10 gene on chromosome 10 and KMT2A on chromosome 11. But also on the same region, you can see that there is a complex rearrangement. There is an inversion within the translocated segment, and there is a deletion in chromosome 11. This is important to highlight and to visualize an optical genome mapping because reciprocal or simple translocation between KMT2A and MLLT10 is not sufficient to produce the fusion product because the genes involved in this translocation have opposite transcriptional direction. The MLLT10 gene is transcribed from the telomere to the centromere, while the KMT2A gene is transcribed from the centromere to the telomere. That's why, in order to bring these two genes into juxtaposition, complex genomic rearrangements such as inversion, sometimes insertions, or even deletions are required. I have several other cases which are showing exactly the same concept. So this patient is 52 years old with bicytopenia and circulating blast. Examination of the bone marrow again showed myeloid blast, and immunophenotyping confirmed AML with monocytic differentiation with the expression of monocytic markers 14 and 64. For this patient, karyotyping showed trisomy of chromosome 4, trisomy of 21, and translocation 1011. In this particular case, the translocation in 1011 is not involving 11q23. You can see it more proximal at around 11q14. And however, we know since KMT2A is involved, there might be other complex rearrangement that cannot be visualized with this resolution in the karyotyping. FISH for the KMT2A break-apart probe showed a normal fuse signal and a signal which is broken apart, I would say, so a red and green signal. There is an evidence of KMT2 rearrangement in this patient. However, OGM will help to identify the exact breakpoint. In the whole genome VO, in the OGM, you can see the trisomy 4 and trisomy 21. And in the circus plot, you can see the translocation 911. So going into the molecule view, you can see that in chromosome 10, the molecule is aligning to the MLLT10 gene, and in chromosome 11, KMT2A gene is involved. However, there is also an inversion within the derivative chromosome 11, and this is again the same concept that you need other inversions to bring the fusion product, to make a functional fusion product. A third case, 49-year-old presenting with lymphadenopathy and circulating blast. The bone marrow was full of blast, and immunophenotyping showed a monocytic differentiation of the myeloid blasts. Karyotyping here showed monosomy 7, and there was additional material on chromosome 10. However, with this resolution, it was not clear what translocation was involved. Interestingly, interface FISH was normal, showing 2 fuse signals. However, in metaphase FISH, you can see that the KMT2A is not in the correct orientation. It's on the P arm of a probably chromosome 10. Optical genome mapping was helpful in this case to show the exact translocation 1011. Here you can see the genes that are aligned in the translocation, but also there was an inversion within chromosome 11, leading to the functional product. A last case, 27-year-old with pancytopenia and circulating blast. Bone marrow was full of blast, and again, this case had a monocytic differentiation of the blasts. Karyotyping showed, in this case, a translocation 1011, and there was a loss of the Q arm of chromosome 14. FISH for this patient again was normal, showing 2 fuse signals. However, you can see that the derivative chromosome 10 having the KMT2A gene on the metaphase FISH. Optical genome mapping showed the translocation 1011, again a complex translocation having inversion within the chromosome, deletion, and the translocation. So from these 4 cases, we learned that a simple reciprocal translocation is not sufficient to produce an in-frame fusion. because the genes, as we said, are involved in opposite transcriptional orientation. This is one challenge. The other challenge, KMT2A rearrangements can be cryptic on conventional chromosomal studies. OGM is very useful in these cases to identify the cryptic translocation, to characterize the exact breakpoint, and to reveal complex rearrangement as they are commonly seen in this type of translocations. That's all for my talk. If you have any questions, feel free to ask.
Great, thank you very much, Fatma. Those last two cases particularly are a good example of why tests like this are so important. Does anybody else have any questions from the group? Yes, I'll ask one. So I know we don't routinely do FISH up front for things like KMT2A unless we suspect it clinically or from the chromosomes. In which case, we'd find some of these, but others we haven't seen. As you saw already, the last two had a normal break apart FISH pattern. And unless you're doing meta-based FISH every time, you're not going to know it's on the wrong chromosome. So labs that would rely on KMT2A FISH to find these may not see them. Do you have a sense of why those didn't break apart? Why was that a negative KMT2A rearrangement by FISH?
So I believe, according to the FISH probe we are using, the whole KMT2A gene is translocated to chromosome 10. I know there are inversions, but the split in the inversion, the split in the genes is very small and cannot be visualized within FISH probes, I believe.
Great, yeah, that makes
Eric.
Yeah.
I actually think what happens is that the piece that moves is actually quite small, and that the flanking probes are actually relatively 5' and 3' of the gene, so they're not actually disrupted. So it's just actually a very small insertion, generally speaking, and you just don't see the break apart because you're not actually displacing any of the 5' or the 3' KMT2A signal.
I think in the few cases we've seen this happen, you can clearly see the whole thing on 10p. And my assumption has always been that the break is, like Fatma said, the break is there, but it's just moving the material to the other side and not far enough away that in an interface nucleus you're going to see it. So I'm not sure if that either makes a lot of sense. Either way, you're not seeing
because sorry, maybe I missed it. Because I thought one of the maps that she showed, you can see there's three alignments, right? The three pieces of alignment. So there's a piece that aligns to 10, a piece that aligns to 11, and a piece that aligns to 10 again, right?
Yep.
Yeah. Yeah, sometimes it's just helpful to get the probe map and figure out where they are and then figure out what's actually happening.
Yeah,
Yes,
thank you, Adam.
exactly.
I think we actually have mapped this out a few times, especially the first time that we saw it, and we did see that it's both pieces of the locus are translocating over onto 10, which is why you don't see that break apart, but it is rearranged. And it does seem to take a bit of FISH workup with some additional probes to really convince yourself where both of those signals are living.
Yeah. Yeah, these translocations, I mean, I'm speaking from some experience we've had with ones we've seen at UHN, and they can be quite different. Like those, so the 1011s they don't tend to be simple translocations. They have to be more complex. So they usually have 2 or 3 breaks. We had one actually that had a downstream component as well. So like there was another break on 10p that was like about 5 megabase pairs away from the MLLT10 break. So, there's a bunch of pieces moving around. So they do tend to be complicated. And back just to Eric's point, of course, that, without having OGM to sort of sort this out, it's really difficult using FISH to figure it all out.
Absolutely agree with that.
Yeah, I think two of these were not seen. I mean, one was clearly seen logically, but then the FISH was negative by KMT2A break apart. One was not seen, and the FISH was also negative by KMT2A break apart. But one of them that looked like a KMT2A rearrangement, right breakpoint on 11, negative by FISH, but positive by OGM. So a couple of these have only shown up because we have OGM. So it's highlighting the importance of having a kind of agnostic test to not have to know exactly what you're looking for every time. Great. Thank you very much, Fatma and everyone, for weighing in. So next up, we have Niven. I think it's a case. Do you have yours queued up?
Yeah. I'm trying to share. Hi.
Hello.
Good evening, everyone. In Israel, it's also now is evening. So I'm Nivin from Haifa in Israel, from Cytogenetic Laboratory in Rambam Hospital. We have been performing OGM tests in the clinic for about 2 years, particularly for hematologic malignancies. And today I would like to share a case. I think it's an interesting case. It's an interesting case where the OGM proved particularly helpful. So let's start. A 50-year-old patient was diagnosed with lymphoma with the positive translocation IgH and BCL2. This translocation confirmed by PCR. The patient received treatment for lymphoma, and 10 months later, we have referred to the cytogenetic laboratory due to clinical suspicion of leukemia, BLL. You can see here by the immunophenotyping test that it's his cells positive for CD19 and for CD10. It's also confirmed support the diagnosis of BLL with no evidence of lymphoma cells. And here you can see the result of the optical genome mapping. You can observe the translocation involving 3 chromosomes, chromosome number 8, 14, and 18. Both translocations were observed with similar VAF, as you can see on the table below the circus plot, which suggested that they may occur in the same cells. However, it's not 100% certain, and we didn't sure in this step if it's also right or not. Notably, OGM provided further insights suggesting that the leukemia resulted from the transformation of the same lymphoma cells with the IgH BCL2 translocation. And this transformation was accompanied by an additional aberration, the c-MYC and IgH translocation. And here, using the genome browser, we can observe the genes involved in both translocation, the IgH with the BCL2, and the second translocation, c-MYC with the IgH. Sorry, it's not need to be like this. Okay. Okay, to confirm our theory that both translocation occur in the same cells, we perform a FISH analysis and karyotype analysis, and the FISH also confirmed that both translocation occur in the same cells and also the karyotype. You can see here, you see my mice maybe? Okay, first of all, by the karyotype, we see that we have a trisomy of chromosome 14. We detected also by the OGM also detect this trisomy, and the FISH detect the same finding. One chromosome had a normal IgH, and the second copy has a split design, and the third one as a deletion of the 3'. The same we can see here by FISH, we can see here in the metaphase, and the FISH also we can observe here the both translocation. The middle one with the 8 chromosome. Sorry about the poor karyotype, but this is what we get in our laboratory for this case. Sorry, cannot I move? Just a minute. Yeah. Okay, so in this case, we report a lymphoma patient whose disease transformed to leukemia. Evidence for both diseases was observed by OGM and suggested clonal transformation. It's very important to mention here that the FISH and the karyotype analysis supported the evolution of the original hematopoietic clone to the leukemia. And we think that it's very important to preserve both the new and the traditional methods in the cytogenetic to solve like these cases. Thank you very much.
Great. Thank you for that interesting presentation. Can you show that? Sorry, I'm a hematopathologist. Can you show that first flow slide again? I may have missed part of
The first step.
the flow
Sorry.
case summary.
Which one? This?
The flow that one right there. Perfect. Okay.
Yeah.
Perfect. So before I ask any questions, are there any questions from the larger group? Because my questions are more pathology related than OGM related.
It's a beautiful case.
Sorry?
I said it's a beautiful case.
Yeah. It's a very beautiful case that help our doctors to understand what happened here. Because when this patient received in Rambam Hospital again after less than 1 year, so the immune phenotyping also observe only the leukemia and the leukemia cells, and they didn't observe the lymphoma cells. So, and the OGM confirmed this, that here we have a case that the lymphoma clone transformated for the leukemia, and not additional clone happened for this patient.
I think we haven't done a whole lot of lymphomas. From my view, I've seen the big advantage is knowing that IGH status of different rearrangements. So we often assumed our MYC break aparts were MYC IGH because MYC and IGH were rearranged. And IGH wasn't with BCL2, but then we do a couple cases and it's clearly not a MYC IGH, it's a MYC something else. And there's some interesting evidence that those are maybe prognostically different. And I'm sure mechanistically the people researching these are very curious about what's actually driving it. We get a couple of cases like this every year in our hematopathology group, and one question I have isn't so much OGM, it's more the pathology. It's was this called a B lymphoblastic leukemia? Do you know? Like a B cell acute leukemia? Because occasionally we see these cases that look exactly like acute leukemia morphologically. But the phenotype and more importantly, the genetics look more like lymphoma, because high-grade B-cell lymphoma can be blastic. So MYC, IGH, MYC, and IGH BCL2 together typically define like double-hit lymphoma. And occasionally we get cases referred to us that are acute leukemia morphologically, and even flow cytometry can look kind of like acute leukemia. They can express some immature markers. But if you do some other testing, like we often will do IGH-based hematocrite mutation testing, we can see they're actually hypermutated, which tells us it's actually a mature B cell. So cases like these, we always kind of wonder, morphologically, it looks like acute leukemia, but is it? And I see Ken has his hand up.
Yeah, so I've seen several cases similar where, yeah, you have a leukemia that has sort of overlapping immunophenotype with large cell lymphoma. And so it's interesting. So yeah, and those cases have all had sort of double hit, or in some cases triple hit, including BCL6 as well as BCL2 and MYC. And yeah, so it's interesting that so I mean, in my mind, they should be considered double hit lymphomas and treated as such, but you know, certainly, you know, I guess it's a debate as to, okay, so should it be classified as a acute leukemia, albeit you know, poor prognosis BALL, and treated in that way, or should it be treated as a double hit lymphoma? Interesting question.
One thing I will say is if this is indeed a lymphoma, the few we've done with OGM, this is remarkably clean for a lymphoma genome. So
That's what I was thinking. It's a beautiful, like, clean case.
but we haven't done a lot of them. So I don't know, double hit lymphoma, is it cleaner? Maybe. Hard to say from my experience. That's great. Thank you very much. Any other questions on that one? Great. Thank you for sharing that. And last set of cases we have Fletcher. Able to share your screen?
Okay, so I'm going to toggle between this PowerPoint presentation and VIA. I would admit that we are, I would say, using the decision trees, but in its infancy. So we're mainly relying on filtering for the moment. So let's start. So our first case is a patient known for CML, which is now transforming to ALL. From the molecular, we knew that BCR-ABL was rearranged. We tried the karyotype, it failed. We did the RSAGH and the SNP array, and since we had these data, we decided to include it in our OGM validation study. And at this time, we built an ALL gene list of 70 genes, which was prepared using the literature. I would say that the next step suggested by Melanie will be pursued in the future. So adding the information regarding their common mechanism of action. So using VIA, we first start by looking at the whole chromosome view, best way to identify the large regions of gains and losses. And after this is done, we will move on to the circus plot to look at the different rearrangements that are present. So as you can see, let me toggle a bit more. You can see at least this BCR-ABL gene and other partners being present here. And we will end up by classifying the regions. So actually, we needed to build up our confidence in what was called with VIA. So almost everything has been classified with tier 1, tier 2, 3, even regions with no genes, benign constitutional regions, and a lot of artifacts. Regarding the AOH and allelic imbalance, we were having the SNP array to compare. And we did find that most of these AOH and allelic imbalances are not confirmed with the SNP array that we are using. So we mostly won't be using the AOH information apart from all chromosome or all arm AOHs. And if we take a look at our tier 1, so we have the translocation 922. That way, we do see that we have a loss of 9p, which has different subparts, and we ended up with a biallelic CDKN2A deletion and NTAP deletion also. So you can clearly see with the OGM coverage that it's a biallelic loss with that case. So if we, and in fact, this was confirmed with the RSCGH. It was what was observed. The second case is an AML, which is secondary to a treatment for a pulmonary adenocarcinoma treated a few years ago. As you can see, the quality is not really good. You can see this addition on the 15, looks like a deletion on 7, and the 16 are a bit odd. Well, maybe an inversion, 16. At the moment where we received the case, the lady was brought to palliative care. So for the standard of care, there was nothing done more, but we decided to continue to work it and include it into the validation study. So that was what was obtained when we take a look at VIA. So we can see the deletion on the 9. In fact, there is an addition on the 8, which is probably what was added on top of the 15. We will take a look at the karyotype after. And if we look again at our tiers, we can see that we have a deletion in chromosome 4, including TET2 gene, and the reciprocal fusion being present. And yes, finally, we have an inversion 16 implicating CBF beta with this one. And for the 8, let's see where it is. So this gain on the 8 Q, we can see on the karyotype that yes, it's really this addition that is present. And let's see. So we should take a look. Well, yeah, it's 8Q. Okay. And for the last case, it was an AML with myelodysplasia. As you can see, the karyotype was showing mainly hypotriploid clone. This is the anucleotide summary that was built up with our SNP array. So you can clearly see the chromosomes that are at 2 copies, 3 and 4. For the chromosome 5, you will see later what is happening and why it's that large. And when we include it into the validation study for OGM, we can see that with the genome new, there is really difficulties with this copy number normalization. So all the chromosomes that should be at 2 copies are not looking correct. So these should be, wow, more than 2. So in fact, the 2 copies are chromosomes 3 and 4, 6 and 7, but they look like being lost a bit. Those in 3 copies, and you can see the deletion 5. So it's really hard with this case to assess the true copy numbers present. So I think that in real life, if we happen again to have a case like this one, we would do FISH on chromosomes that we suspect that are diploid to confirm the copy number and then assess the rest. So it was really difficult for this one to understand what copy numbers were present. I don't know if other ones have been having cases as difficult as that to interpret. So it's mostly this for me. Thank you.
Great, thank you very much. I really appreciated the live view of VIA and how you navigated. It's still a lot of experience with, but I can definitely empathize with the copy number calling on those non-bytes.
Yeah.
I see now Melanie has your hand.
Yeah, so I have a comment and a question. So the first comment, if you go back to your VIA data for both cases where you have like confirmed gene fusions, if you look at the SV score, you'll notice that they're way below the 20 threshold recommended by Bionano. So I think the first one, the 922, was around 13, and the second one was around 9 and 5. So these are the cases that prompt, like we personally use a threshold of 5, and even then, like in rare occasions, we have to dig out like one in the poorer cases, we have to dig out one of the fusions out of the low quality bin, but we have set it up at 5 because we've noticed that we can have much lower quality. I personally, don't know if how they came up with that 20 threshold, whether or not it's simulated data or not, but in real life, it's actually not working well. So I thought this was really good to illustrate. And my second question, my second point is a question about your triploid case. Have you looked at whether or not there's allelic imbalance that is called that could support slight changes in copy number for the chromosomes that are gained versus, and also the absence of allelic imbalance for the chromosomes that are not gained? Because I know in VIA you can decide not to see allelic imbalance and AOH that correlate with a copy number, which cleans up the view. But when you have a doubt regarding diploidy or whether or not something is gained, I think it's actually valuable to look at that data. So that's something I would encourage you to look at in VIA.
Yes, I will take a closer look at that after. It's something that we have not been working on with the allelic imbalance and the AOH. But we were at least having the corresponding RCGH data to confirm it. And we recently had an ALL with the hypodiploid and tetraploid clones. So I'm looking forward to see how it will look like into VIA. So it should be coming pretty soon.
I would love to see that because we haven't had a case of mask hypodiploid in our core. We have hyperdiploid BALLs and we have one tetraploid AML, but we haven't had any cases of mask hypodiploid. So I would really love to see that data if you want to share it at a next meeting or at other type of venue.
Yes, sure. And I will probably share another case. So we had a PGGFR, was it alpha or beta, that was rearranged. So yeah, we're going to share it. It's pretty rare.
Great, thank you. Yeah, we've had our experience at matched hypodiploid and it didn't perform well. We didn't use VIA, so that's one point against us on that, but it tended to call a couple gains here and there, trying to normalize to what it saw. So we'll maybe push that through VIA and see how it performs, but I suspect it won't be a whole lot different. I see Kathy has a hand raised.
Yeah, Flesher, thank you for your presentation. I agree with Melanie about the AOH data, but I'm not sure if you're aware, in VIA, you can actually recenter your data. So if you know that one of the chromosomes is actually 2-copy, you can use that to make it 2-copy and everything else will shift accordingly. So that'll help you look at your data, but I would also look at the well, I guess they're not really SNPs in this case, because we use this for our NGS panels to look at our copy number, and we use the SNP data to help determine if the tracks are split or not, to help you determine which are even copy numbers and odd copy numbers.
Thanks, Melanie and Kathy. I will discuss with Bionano to find out how to do it.
Great, thank you. I had one question about your first case, the CML. I saw there's a whole bunch of stuff going on in 3Q. Did I have a meet army arrangement in there or no? Occasionally see the CMLs transform and throw those out.
Okay. Yes, on the 3Q, we were having a fusion, which is a fusion. Let me see. It was this one. So it was a fusion with ATR. So ATR and another lock gene. We were able to see a bit of literature on ATR fusions, but we're not really confident to call it being something else than tier 3. But it was true. We did look at the data into Access, and the fusion was really neat, really clean. So without doubt, ATR is rearranged in this case.
Great, yeah, sorry, I thought it looked a little more distant than it was. The other question I had was more for the broad group. How are you guys handling these, so specifically the ALLs that are hypodiploid? I think that suggestion of normalizing to what you know is diploid is great. But I guess the question we always have is, are we going to know it's not diploid? And maybe the AOH tracks being different than you expect is a good hint that it's not diploid, but how are people who are using OGM for ALL handling that problem of relying on OGM to give you some kind of ploidy status that you can count on, or are you just doing FISH and everything, or array?
We will reflex with at least 2 FISH on chromosomes that we think are diploid and see.
you know that the pattern, like there's a known pattern of chromosome gains and quote unquote losses in those masked hypodiploid. And so I think if we have a hyperdiploid with that has double trisomy 410, we would not be overly concerned, because if you look at that pattern, like I know COG, Children's Oncology Group, put out data many years ago and have a retention of chromosome 4 in those masked hypodiploid or masked near haploid is extremely rare. So if we do have a trisomy 4 in a hyperdiploid with or without chromosome 10, we're not overly worried. But if we do have a hyperdiploid that doesn't have double trisomy 410, but does have gain of chromosome 14 and 18, this is where we're more concerned. So we always look at the back in the day when we didn't have VIA, we would run we would always run the RVA on those hyperdiploid BALL. We also ran the de novo to have the AOH information. But now in VIA, with the ability to see the BAF and have the AOH allelic imbalance, we're not running the de novo pipeline on those. But part of the degree of worry is based on the pattern of hyperdiploidy. So there are some good papers out there that have statistical data on the chromosomes that are retained in those versus lost. So it's also based on that that we interpret them.
Great, thank you. Yeah, the few we've come across, because as you're saying, they're rare. The copy number, mostly gains we end up seeing, are very inconsistent. So even the things we know are gained aren't always called as gained, just depending on the subclonal status of them. So we'll see gains of things we know are gained, but then some things that were gained weren't gained by OGM. So we get confused about what exactly it's doing and how it's determining which chromosome to call gained and not gained. Great, that's very helpful. So great cases, everybody. Thank you very much for sharing those. I'm sure we'll have more in the new year. There's no shortage of interesting cases of OGM. So I have a quick run through of data I showed at ASH that I can share, and then we can go to kind of an open discussion. I know EQA is a topic of interest we can try and touch on, but it may be something to regroup in, again, the group that was interested in this before, see where we are, how we can move it forward, because I know that is a growing need. So great cases. This is the screen is sharing, I hope. So this is data that I think I haven't showed to the group here before, but won't be any surprise to people who are using OGM. This is data from our validation that was presented last year at ASH, and using a cohort of about 69 cases that were selected, retrospectively evaluated by OGM, we observe obviously a huge number of variants with OGM. So if you're filtering just against population, the average number of variants median is around 53. When you begin filtering things out with pan-cancer gene lists, you end up much lower, and using indication-directed filtering, you end up obviously in the range of, 3, 4, 5 for most cases. Occasional cases will have these huge numbers of variants, but that's largely copy number calls that are fragmented. What we found particularly interesting, and always do, is the number of cases that have changes in their classification, so pathologically, or changes in their risk stratification based on OGM. So if you look across the whole spectrum, around 60% of cases had additional findings that were, sorry, 50% had additional relevant findings, 25% had clarified or cryptic drivers, and about 10% had a classification change. But when you split that out to things like AML, we're seeing in the validation cohort, about 13% of cases had a change in pathologic classification, about 20% had a driver we could now see, and 40% had other things we couldn't see before that we know are somatic and cancer associated. So these are largely the ALL microdeletions. What that looks like when you look at the reclassification scheme is mostly patients being reclassified as higher risk. So normal karyotype AMLs being shifted is the bulk of it, where a normal karyotype goes to NUP98, for example, rearrangements, which we know are cryptic, KMT2A rearrangements, which can be cryptic, as you saw with Fatma's presentation here. Occasionally things like AML MRC, where you have deletions of myelodysplasia-defining genes, or in one case we had a del7q that was not seen because it was an unbalanced rearrangement with a 10q that looked quite similar on low resolution karyotype. Those had a couple cryptic MECOM rearrangements pop out of these AML MRCs that, as what you'd expect, behaved relatively poorly. Importantly, none of the critical things that we know we can usually find, like conversion 16s, APLs, base variables, were reclassified. I think the most exciting thing from my perspective is always the new stuff you see. The cryptocurrency one, MECOM, wasn't anything novel, but is something that was invisible because the karyotype basically failed in this case. But things like this FLT3-PAN3 dysregulation have been reported rarely in the literature, but we're beginning to see them much more frequently in BALLs. And maybe there's something to them that we can make sense of once we have enough data. So that's a validation cohort. What I just wanted to show you is the kind of 1 year post-implementation review acute leukemias at VGH. So we've been doing this test since November 2023 now for all newly diagnosed acute leukemias, ALL and AML, and plan to soon expand that out a little bit to chronic myeloid and other indications. But over that 10-month period pre-ASH, this is not updated data, we genotype about 90 people with acute leukemia, most of them AMLs, and a proportion you'd probably expect with ALLs, mostly B, a couple of T, and then one rare mixed phenotype. OGM worked in pretty much everybody we had sample for. The 2 who it didn't work, it would probably as expect work, didn't work because we had poor quality specimen. So low cellularity aspirate or no aspirate at all when we tried the blood, which was hypocellular. On average, we're able to get a result out within about 11.8 calendar days following the sample procurement. So that includes weekends, and that includes the early lead-up period where lab familiarity with this test was fairly low. What I think was interesting was that the same kind of data come out from the 1 year post-implementation on non-selected specimens. So here are cases that had a variant we could find either by OGM or karyotype. About 15% of AMLs were reclassified pathologically. 19% had a change in risk classification. And for ALLs, we saw less pathologic reclassification. And that's possibly just a feature of the cohort being largely B-ALL with B-serial arrangements. But a significant number of changes in perceived risk per leukemia BMT, where they're acting on these IKZF1 deletions that probably have some mixed evidence to them. But here, the group feels they're higher risk. TALLs, we find a lot more variants and find the drivers we could never really see before with our old panels, which is pathologically interesting, from a research perspective interesting, but as you're probably aware, doesn't really change the stratification of the patient's risk right now, or doesn't change the WHO diagnosis because there's no established entity other than the typical KMT2As that defines a TALL entity. What was most interesting to me, again, in this cohort was that we're seeing a lot of previously undescribed drivers that were either cryptic or just not understood in the literature right now. This case, for example, is an AML that has a very small inversion 3 that was not seen on banded chromosome analysis, which doesn't involve the typical GATA2 MECOM loci, but involves MECOM in this TRIM59 IFT80 locus. And this was paired with an ETB6 NTRK3 fusion. This patient performed about as well as you'd expect for a MECOM rearrangement and had pathologic features typical of MECOM rearranged AML. The second case, I think I showed at the great user group meeting last year, was a person who had an incidentally identified MDS from occasional circulating blasts in her blood and a normal blood count otherwise. But karyotype showed monosomy 7 and nothing else. With OGM, we see there's this tiny little insertion of the MECOM locus into this AL58969.3.1 locus, which again, we confirmed by FISH. This is a MECOM insertion. And again, this patient pathologically, in retrospect, fit with a MECOM rearranged high-grade MDS, as we might call it locally, and again, behaved about as well as you expect a MECOM rearranged AML would. So she was upgraded from MDS to AML MECOM rearrangement based on this OGM and moved to transplant pretty quickly. Lastly, these NUP98 rearrangements we're seeing much more frequently than we were before, probably because we weren't doing FISH before. But things like NUP98 HOXD8 rearrangements that are, again, undescribed and do change this patient's diagnosis and risk stratification. This is the limited set of the data we have so far. We have a lot of interesting cases to pull out for future meetings, but thought I'd give you a quick recap of, again, the rates we're seeing of change in diagnosis, change in risk certification, and just highlighting how important testing like this is to have up front. So any questions? I see Fletcher.
Yes, thanks a lot for your presentation, Eric. I'm a bit surprised by the so high change in risk for BALL. Well, I know that we had only 5 cases in our validation cohort, but the risk didn't change for any of these. Yes, and well, in fact, clinically, we're doing the RECGH and the our fish panel. Well, we do the karyotype, but it doesn't bring that much to the story. So there was no risk change for us. Is it because of that?
So the big thing that I think differentiates our centers is we don't do array for ALL. So what we've learned is the fish probe we're using for IKZF1 was quite poor. These lesions are often small, and OGM will find them, where fish often won't. So if you look at the literature reported rates of IKZF1 deletion, they're much more common than we were seeing. And now we're seeing them, is essentially, I think, what the crux of the matter is. And I know there's conflicting evidence on IKZF1. So we tend to report it with that conflicting evidence. The most recent UK ALL14 saying there's no independent evidence, but our local clinicians still feel there's enough of a conflicting evidence base that it does weigh in, at least on their decision making.
Yeah, we do see those IKZF1 plus profile. And you had also one case with a RUNX1 deletion. Was it like an exonic deletion, something like that?
Whole gene.
Whole gene? Okay.
Whole gene.
Okay, thanks.
Hi, Melanie.
I think it's also, just to follow up on Flechel's comment, I think it also depends, like your cohort, these are adults, right?
Adults.
BL. So obviously we have a mostly a pediatric cohort. So I think it does affect, obviously, as we all know, the proportion of different abnormalities varies depending on age. So I think depending on the age of your cohort and whether or not you have some high-risk patient in there as per COG stratification or not, it may influence the data. I assume were most of your BLLs BCR-ABL1 positive, Eric?
30–40%. Yeah.
Okay,
So
yeah.
the majority tend to be.
Yeah. So we're obviously in our pediatric cohort, we have very different data. So I think that's also something to consider. It might be interesting not only to look at, the reclassification of AML and BALL, but also sort of try to stratify it a bit by age group, particularly if, like maybe centers that are doing OGM on these should like band together and publish data, that's something like that. That would be interesting because we certainly have found not necessarily WHO defining abnormalities in our BLL cohorts, but we have found like a FLT3 PAN3, we have found some different, some of those very rare ones. So definitely would change the, if not prognosis, would potentially change management in terms of what therapies these patients may be eligible for.
Great, thank you. Yeah, the FLT3 PAN3s, we found a couple as well. So I'd be interested in what you've seen with those. The first time we saw it, we kind of went, that's weird. And then saw another 2 or 3. And there's some good literature about exactly what they do, which is pretty cool. What to do with them, I don't know.
Yes, I mean, we could, maybe we can collaborate. We have one so far. So maybe we can collaborate and.
Great. Yeah, the ones we've seen tend to be very subclonal,
no, ours, I can't remember, but it was a high volume. Like we didn't a high proportion. Like we didn't see anything else that was really clinically significant in that case.
Interesting. Great. Yeah. So that's definitely something I think we should all talk about in the new year or going forward is ways to collaborate in the community, because we all have data that we can all work on independently, but working together makes a lot of sense, and consolidating that data and learning lessons from each other.
Yes, and I think it would be interesting to discuss strategies on how you confirm some of these findings, because some of them can be easily like confirmed by FISH or karyotype or microarray, but some of these are very challenging. And so if one center has the ability to do it, perhaps we can collaborate, all of us, and help each other out, because I think that part of it is you see things and you have to confirm them, but it's not always that easy to do.
Great. Thank you very much. Any other questions or comments before? So I see we're approaching 10:20 or 10:40, 20 minutes to whatever time your meeting ends. Suppose one thing I want to touch on before we consider wrapping up early, unless people have items that they to discuss, are first off, a reminder, if you haven't already registered for this and you are interested in being on this listserv, please send an email to this email address. I will send this out to people who have attended today, so you can add that to your to-do list if you'd like. And I know we've had probably 20 or 30 people added to that list, but it's been quite quiet to date. So if perhaps in the new year, we can try and kick that off and get some asynchronous discussion going where it's appropriate, that would be, I think, a great learning opportunity for everyone and a good tool for us asking one-off questions like, what is this? Or what do I do with this? EQA is a topic that we've talked about before in kind of dedicated meetings, and there has been discussion about centralizing that in some way to a couple of centers with case sharing. And the main barrier that came up from our institution's perspective was the privacy barrier of this is patient genomic data that you'd be sending off-site, and at least our privacy office wanted to work with that a little bit before we could move forward. I don't know if others have had a chance to set something up or have additional discussions with their privacy offices or equivalents.
We've been in similar discussions at our institution too.
So in the meantime, hopefully people at least are adopting it more widely, and we're seeing at least opportunities locally to share that are established. So maybe that's a stopgap until we can sort of.
We've signed up for the Gen QA survey, but it still hasn't, I don't know if anybody's heard anything updates about that yet. Not sure. Do you have feedback?
No. In October, we did a molecule file exchange with one of our other Quebec hospital. We had only one thing before and with this molecule file, which was the diagnosis of the patient. We just find that it was a male or a woman, but you don't really see something that you could identify the patient. A monosomy 7 deletion 5q, a translocation involving MICOM. It's not that a breach of confidentiality to us, so it was not raised in our center.
Yes, and no, operationally for us, at least, it was that was the question: is it identifying enough? And our health authorities' policies are very very strict on what is patient data, and even a CBC result is patient data. So someone's hemoglobin level and white count and neutrophil count is considered protected, right? So at least from VCH, that was the limiting factor, was what is considered identifying enough. It was quite a low bar to meet that. So our operations felt that we had to go through the privacy office for any kind of sharing arrangement. Great. So maybe we can reconvene on that and update where individual centers are at. Hopefully we'll have something to move forward with in the new year. This commercial EQA maybe will eventually be available. And the other item we kind of touched on today was opportunities for collaboration. So things like collaborating on gene lists via frameworks or decision trees, I think is a great thing to discuss and plan for. So maybe we can plan to discuss, Susan, Adam, myself, Jose, how to spread those emails around and just be in touch by email, see who's interested in what topics, who's interested in research collaborations via gene lists, everything, whatever it might be, and try and band together to get some of that addressed, especially now that OGM is growing in Canada.
agree.
Great. Well, thank you everyone for attending today. Again, remember, if you have good cases come up, save them. We'll ask you for more in the new year. And please feel free to reach out anytime to the OGM COP Gmail address. I do try and monitor it, but if you don't hear back from me within a couple of days, please feel free to nudge me again. But I will make sure we send out this listserv email at the very least today, so we can get people on there while they remember.
And don't forget to mention, Eric, that the recording will be posted, and you got a follow-up email from U of T about that as well. So if anybody couldn't make it today, or you have members in your group that would benefit from the presentation, you're gonna be able to watch the recording later.
Thank you, Adam. Always forget that part.
Oh, and do the survey too. There's a survey for CME. If you want CME credits, you have to complete the survey. You'll get an email, and there's a link in the chat.
Thank you very much.
Happy holidays, everybody.
Happy
Happy
holidays.
holidays. Stay safe.